Hello, my name is Christian Tischer. I work at EMBL Heidelberg, and I will speak to you today about MoBIE, which stands for Multimodal Cloud-Based Big Image Data Sharing and Exploration at Fiji. And I would like to thank the organizers of this conference very much for giving me the opportunity to speak here. So what is MoBIE in a nutshell? So it's a big data viewer-based Fiji plugin for multimodal big image data browsing, and there is also an update site called MoBIE to install it. I would like to mention that Big Data Viewer was not developed by us, but has been around for quite some time, but turned out to be very useful for what we want to do. Then on top of this, MoBIE is a set of specifications and implementations for cloud-based big image and metadata storage, and you can find most of this on the MoBIE GitHub.io website. So why did we do all of this? So the challenge we had was, how do we visualize and browse multimodal image data? And here you can see such an example data set. We have two electron microscopy images. One is a single 2D slice transmission EM, and then a much more high resolution 3D tomogram. And then also we have fluorescence data that we would like to view. And as you can see, these images not have only different pixel sizes, but also different dimensions. So viewing them in a standard viewer like ImageJ is not possible, because they're all the images that you want to overlay should have the same number of voxels and dimensions. However, using Big Data Viewer, this is readily possible. And the trick here is that the data model is as follows. So you store your voxel data as normal on disk in a grid, but then on top of this voxel data, you also specify a transformation, and typically it's an affine transformation, how to go from the voxel space into physical space. And that's the space that's shown in the Big Data Viewer window. And there you can put really all the scaling, translation, even shearing if you want, and rotation. And then you can view these three data sets on top of each other without the need to resample any of this or resave it. So you can just project them into the place where they should be. Another thing we implemented on top, which is not standard in Big Data Viewer yet, is a blending mode that we call occluding. So this is nice for EM, for example, here where you have the high resolution tomogram, you don't want to see maybe the low resolution transmission EM. So this is in fact done by this occluding mode, where you now only see the tomogram in this place, but the fluorescence you see everywhere. Another big advantage of this idea is that if you want to now, for example, improve this registration, right, you have to register obviously these images, that this does not require you to resave the images, which can be very annoying if they are very big, but you only have to change a few numbers in this little text file, which specifies the transformation of where to put it. So this is also a very big practical advantage of this Big Data Viewer image data model. Okay, so the other challenge we had is that our data was quite big. For example, the data set you see here is a 3D volume of a Platinarés worm, which is 2 terabyte of image data, or even 8 terabyte if you just store it naively with all the black pixels. Now, to view that, you really want to have a chunked multi-resolutions like Google Maps-like storage. And the idea of this is the following, and I try to illustrate that here. So you save your data not only once, for example, here on the left in a low resolution version, but also in maybe several higher resolution versions. So here you see the pixels are much smaller, and that's because it's stored at a higher resolution. That means if you now zoom in, you start loading from a different image, sort of, yeah? So the higher resolution one. But if you still had to load the whole image, that wouldn't really help you, right? So the other big trick is chunking. And the chunks I tried to illustrate here with the thicker lines. And everything that's within one block of thick lines is stored on disk very close to each other, such that can be loaded very efficiently. So the trick now is, if I zoom here in the middle, I only load the few chunks that are actually needed for the data that I want to look at. And if you implement this, you can very smoothly browse terabyte-sized images like on Google Maps. So now the question is, how do you actually implement such a file format? There are different ways. One way we support is the OME-ZAR way. And there the concept is quite easy. So you just say each chunk is a file. So for example, how this would look like if you would store a 5-dimensional data set like this on disk, like with time, channel, set, y, x, then here actually the first folder specifies the resolution layer. So this would be the highest resolution layer in this case. And then all the others are the dimensions. So this is 0 chunk in time, channel, z, y. And then here we go to the 0, 1, 2 second chunk in x. And that contains all these pixels here. Okay, so if I want to view this, I just have to load this file. And as you can see, for example, here, this file is very small. So if you have very many small chunks, you can very efficiently just load a subset of the data. What's really great about this format is that it maps very well to object storage, because you can then basically just say each file is one object in such a cloud store, and then you just download all the objects from the cloud store that you would like to read. But we also support other file formats. So this is the one I was talking about. We also support a very similar file format developed in the Saalfeld lab at Janelia, which is the Big Data Viewer N5 file format, which works exactly identical. And then we also support the Big Data Viewer HDF5 file format, and some of you might have heard of HDF5. So this is also very suited for this chunked storage, and you can also store their multi-resolution pyramids in a convenient way. However, accessing small chunks from an HDF5 file in the cloud is a bit more tricky, and we didn't get it working just like that. But I heard somehow that this should become possible now, so maybe it will be working, but we right now don't support this. So we support the HDF5 format only on the file system currently. All right, so, but a MoBIE project is more than just images. So here you see the images. It's a bigger folder structure comprised, so the entry point is a so-called project, and then there are essentially JSON files that specify more. So we can have several data sets, and each data set is again specified by a JSON file. And we can also have tables, and I will get back to that later. And also here now we support different ways of storing this. So the easiest would be you just put all of this on your file system, right? That's probably typically how you would start. Then if you want to share with collaborators, you can just copy all of this on an S3 object store. And this bucket symbol here is because in S3 object stores, one speaks about a bucket where you put many objects. But we actually support also something else, and this is what we mainly use in practice, which is where all the big images are in the object store, but then all the smaller files, which are the metadata and the tables, we put on GitHub. And this is, we think, very practical, because we quite often actually changed JSON files and the tables, and this is nicer on GitHub because you have also versioning, and it's just a bit more accessible than the object store. So this is a modality that we currently mainly use. But you can't put big images on GitHub, right? That's why we have them in the object store. Okay, how do you open such a project? Well, you just start Fiji and you type in either a GitHub web address or the main folder on your file system or the main S3 bucket, and then you just press OK. And the one that's here, actually, I put it for a reason, because this is what triggered the whole development of MoBIE. So this was work together with the Arendt lab at EMBL and also Schwab lab and Kreshuk lab on a Platina Reis data set. And this is published. And this was very big, right? Many image sources adding up to a few terabytes of data of different modalities, and we really didn't know how to share this data with other people and how to browse it ourselves. And this is actually what triggered the development of MoBIE. But then MoBIE turned out to be useful for also other projects. So now it's actually used for many, many more things. But this was the initial motivation. So if you hit enter on Fiji, then it will look like this. So you have a main user interface where you can select from the different image sources, and you can then also change color and brightness and so on. What you see here on the right is now the electron microscopy on top. And on top of this, 2 light microscopy images, which indicate gene expression patterns. For example, here a gene that's mainly expressed in neurons and another gene that's mainly expressed in muscle cells. And as mentioned earlier, one can easily overlay them even though they have completely different resolutions. So another interesting thing is that this is actually all 3D, and also the chunking is 3D, which is important because in a normal viewer now, let's say this is the slice you look at, you could only go up and down in Z, right? This is what you're probably used to. But this is not the best orientation to explore this animal. Yeah, this has a much more natural axis from its biology, which is this view, where the head is on the top and then you have the arms on the right, and here you have the neurons. And the nice thing, and this is also enabled essentially by big data viewer, is that you can just with dragging your mouse and holding the left mouse button change the angle that you view your image in 3D, right? So this is very important. Okay, then we have also a bookmarking. So for example, if I go back to the slide, if you found this very nice orientation, it's of course good to store this and share this with your collaborators, because finding this exact orientation back in 3D can be very difficult. So one can essentially just bookmark the current view and then save it. And what we did is, for example, for this publication, we tried to make all figures a bookmark so that you can look at this in Fiji in live rather than just looking at it in the publication. So here you see this. And for example, now let's look at the epithelial cell segmentation bookmark. If you click on this, something like this will pop up on your computer. So first of all, it will have zoomed in here into a few epithelial cells. It will also have loaded the segmentation for these cells, which were, by the way, done by Constantin Pape using some deep learning approach. It will also trigger viewing these selected segments in 3D, which is something we also support. It will load an associated feature table, where for each of these segments you can have several measurements. And it will also open a scatter plot where 2 of the columns in the table are selected so that you can also explore these features in a scatter plot way. What's important to note is that the coloring model here is shared between all these 4 views, so you can easily find an object back. And also, if it's all interactive. So if I would click on a table row, it would highlight it in all the other views. If I click in a dot in the scatter view, it would highlight it and so on. So this is a fully interactive way of exploring this data. Then what we also support is different coloring modes for the segmentations. So what I had on the slide before and what you see here on the left is a so-called random coloring. So this is probably the best if you just want to see what are different cells. But maybe you want to see something else. So for example, here now we color by the gene cluster column, which is something that was done by Valentyna in Anna Kreshuk's lab. And here all the cells should have the same number and then the same color if they correspond to the same gene expression cluster. So you can color here by any column of the table, and like this. Also, I think it's very convenient for biological discovery. We also support annotation of these segmentations. So in the big data viewer window, you can just click on image segments, for example, here the different cells. And then we have here a user interface where you can freely annotate them with whatever text string you would like to have. And this would then simply trigger the creation of a new table, a new column in your table where these annotations are stored, and then you can save it and then share it with your collaborators. This is used for biological annotations, but also can be nice for technical annotations. For example, let's say you found a cell that was not nicely segmented. You could annotate it like this and then, you know, share it with the person that could maybe fix this. Then something else that we added quite recently, in fact, is something we call the grid view. So the idea here is that if you have like a sort of consistent image that always has the same size and it makes sense to just browse many of these next to each other, then it's nice to just lay them out in a grid. So one example would be, for example, high throughput fluorescence microscopy data. This is something we also are supporting soon, I think. But this is in fact another example. So these are many 3D tomograms that were acquired in the publication that is mentioned here at the bottom. And you can now quickly like this browse through all of them. And the other nice thing is because you have a table with annotations in it, you could also, for example, now I colored all the ones where there is a Golgi visible, and that's also a nice way to get an overview. Okay, how many of them do I have? And then also you can browse to one of them, zoom in, and then basically look at just this tomogram. And I would just like to mention that this is all in 3D. This might look a bit 2D here, but you can also just at any time look at these tomograms from the side if you want. Okay, then how do you actually create such a project? So one way is in our Fiji plugin. We have a way where you can add images one by one and then populate your project. And then it will automatically create for you all these JSON files and the corresponding folder structure. Since you have to do this manually, this doesn't scale very well. That's why we also have a Python library where you can do all of this automated. So the best would probably be to contact us if you want to do this, and we are very happy to help you. So finally, as an outlook, so what I was talking about today is here the things on the left, right? So MoBIE is a Fiji plugin and a set of specifications for this big multimodal image data. So what we try to do, and we collaborate actually very intensely with the OME consortium, we try to put as much of this as possible and as makes sense into this OME.zar format that I was talking to in the beginning. And so what I should say is this format is actually under development, right? This is a new thing. So this is basically the idea. We as a biological imaging community, we really need a good file format for big image data, and that's also cloud compatible. And this OME.zar is at the moment what we think is maybe the best way forward. But the metadata specification, for example, is really in its infancy. So there is still a lot to do. And basically our idea is also a little bit, let's try out what works here for us in MoBIE and then sort of suggest, can we put some of these things as a community standard? And really the vision is, if some of this works, it would be fantastic, because then you can take this file and not only view it in Fiji, but also in Python, in Apari, for example, and in all these emerging web-based viewers, right? So the vision really is you save this once and then you view it everywhere. So by the way, if you're interested in such like this, you developing things like that, you could contact us. We have probably soon a position open to work on this. In addition, I really would like to encourage you to get involved in this process. So if you just Google OME NGFF, where NGFF stands for Next Generation File Format, you should find a bunch of entry points to join the discussion and also the process of making this file format such that it's good for you, right? This is really your chance to get involved here. Okay, then I come to my acknowledgements. So first of all, I would like to thank the MoBIE core team, Constantin, Kimberly, and now also Ekaterina, an intern who helps us, which is really great. So she actually helps with the OME.zar integration, and that's already working now. So thank you very much. This is fantastic. Then I would like to thank several groups at EMBL, the Advanced Light Microscopy Facility, the Electron Microscopy Core Facility, the Kreshuk team, and the Arendt lab. So as I said, in the Arendt lab, this is sort of where this whole MoBIE project was triggered, because we needed a way to view this big Platinares data set. And there are many members in these groups that I would also like to thank, but the slide was too small to put them all on. And then I would like to thank George Moore, who heads this OME ZAR development at OME, so we collaborate very closely with him. And then finally, I would like to thank the Chan Zuckerberg Initiative for very generous funding. And you for your attention, and I'm happy to take any questions that you may have. Thank you.